All right. Good morning or good afternoon or good night, everybody. Yeah, it's my great pleasure to welcome you back to the VMOL seminar today. We have Helena Russo being our speaker, and yeah, it's my great pleasure to have her talk about the CMMC and the knowledge base they're building within the GNPS environment. So this is a very exciting developments. But before she gets started, just like a very brief background. So Helena is a chemist. She did her undergrad at the State University of Campinas in the state of São Paulo, and then also a PhD in chemistry at São Paulo State University. During that time, she spent already a couple of months in the Dorstein lab as a visitor, and then now also came back as a postdoctoral researcher to Peter's group. And yeah, I think she's been doing some monumental work. Some of you may have heard about, and yeah, I'm very much looking forward to hear more about the CMMC Knowledge Center. So yeah, the stage is yours, Helena.
Hi, everyone. Thank you so much, Daniel, for the kind introduction. It's really my pleasure to be here participating as part of this VMOL seminar series. So thank you so much for the invitation. Today, I'm going to be talking about the Collaborative Microbial Metabolite Center and get into more information on how you can reuse this information of the infrastructure that we are building. Well, but to start, I believe that everyone here in this audience recognize the importance of microbial metabolites, be it for the study of natural products or for microbiome-related studies and implications in human health. But as many of you know, in the untargeted metabolomics world, here represented by this black screen, only a small percentage of the metabolites that we can capture can be annotated, only about 10 to 15%. And of this 10 to 15%, only about 1% that we can really link to the microbes that can be producing or modifying these compounds. So we are actively working on approaches to improve these numbers. Here, I'm just giving a very brief example of a data set that it's a subset of the American Gut Project, which is a huge project that comprises thousands and thousands of fecal samples from the United States, but also a big sample set from the UK and also Australia. So there is this public data set that comprises about 3,000 samples. And when we do, for instance, classical molecular networking with this subset of data, we get about 51,000 nodes. And of these, we have about 9% of this spectra that matches with the public spectral libraries that are available in GNPS. So it is very close to the estimations that I mentioned previously. And then when we go to the annotation table, we will see, wow, we have 4,700 annotations for this data set. This is amazing, isn't it? But well, we must consider that in this public libraries, such as the GNPS public libraries, we're going to have a very heterogeneous type of data. So in these libraries, of course, that we are going to have human-related, host-related molecules. We're going to have microbial molecules. We also have food-derived molecules, plant molecules, drugs, and also pollutants such as other human-made molecules, such as pollutants, pesticides, and such. So if we are interested in exploring more deeply, more specifically, the microbial-related molecules, we end up with a big question mark in our head, because we don't know what is the percentage of these 4,700 molecules that would be microbially related. And what you can do, of course, is do a bunch of literature searches and Google searches to see, okay, which of these 4,700 molecules are microbially related? And of course, that you will all agree with me that this is very time-consuming and it's not an optimized way. So, well, the struggle is real. And it is so real that the NIH put up a big call combined with 7 different centers, recognizing how difficult it is to mechanistically understand the microbiome if we don't even have a centralized knowledge base in which we can capture and reuse such information about microbial metabolites. So having that in mind, we created the Collaborative Microbial Metabolite Center, aka CMMC. And this center basically, as part of this center, a knowledge upload portal was created in which all the information and knowledge about microbial metabolites would be captured by this upload portal. And well, as important as capturing this information, we are also working on ways to reuse this information in downstream analysis. And to enable this, of course, that we are providing several trainings in the form of workshops. We are also working on the documentation. And well, we are also managing the curation, storage, and ways for knowledge enrichment of these microbial metabolites. And well, who is going to be populating this knowledge base? There are a lot of other grants that were awarded as well that are connected to this big initiative. This for the CMMC. So these grants, they are basically working on microbial molecules in many different approaches, and all the discoveries and all the data that they are going to be generating are going to be populated, are going to be deposited in the knowledge base. But of course, that this is not the only source of information that we will have. We are also, of course, opening up for the community to also deposit microbial metabolites that they are investigating. Okay, so the microbe mast, which was already another seminar that was given some months ago. It's part of the VMOL seminar series, is a part of the CMMC efforts, and we also have the CMMC knowledge base, and further down, we also have the network enrichment analysis, and we are going to be focusing on the knowledge base and the network enrichment today. So right now, I'd like to, of course, point you to the CMMC website. So here you're going to find a lot of information. You'll see here that there is a tab for the documentation on how you can deposit data, how you can reuse that. Of course, I'm going to go through here in this workshop, but if you are looking for more information, more detailed information, you will find it all here in this website. And well, I will go to the hands-on part now. It was very brief here at the beginning, but all of the step-by-steps that I'm going to be following are in this presentation that I'm also going to share with you. So let me stop sharing for now. And yeah, so I created this Google folder with important information. Daniel, if you can please post on the chat. Here I put my presentation, the PowerPoint presentation that has all the step-by-steps of everything that I'm going to be doing here today, in case you want to check that out later. I also created this other, this Google Docs here that will have some important links later on we will use some. And I also, I'm going to go through the other files later on. But well, the very first thing here would be to go to GNPS2 and log in. Here in that case, I'm already logged in to my username in GNPS2. And well, for us to deposit information in the knowledge base, we can go here to workflows. And then what we can do is type CMMC. And then you will see that there are two workflows here that are part of CMMC, and these are the two ones that I'm going to be showing you today. So right now, I'm going to show you the CMMC deposition workflow. So we can click and launch workflows. And here we are going to have basically like a form in which we are going to be inputting all the information that we have about the microbial molecules that we want to deposit. So there are two ways that you can do this upload. You can upload as a single upload, so one molecule at a time, but you can also do that as a batch upload. In that case, I'm just going to be to mention briefly what each field means. This is not really a hands-on part in which I'm going to be depositing information, because of course that it requires us to have prior knowledge on what we want to deposit and such. But I hope that this clarifies a lot of things of the step-by-step for you later on if you want to deposit this data. So the batch upload, in case you want to deposit, for instance, 10 compounds, 20 compounds, you can just copy this tiny URL here in your web browser, and then you will see here that there is, it's basically a Google Sheet that you can make a copy out of that, and then fill out the information. And basically each column here is a specific field in that form that we have on the upload portal. So coming back here, the only thing that is really mandatory for now for you to be able to upload any data into the knowledge base is to have MSMS spectrum. So it's the only mandatory thing. You need to have a fragmentation spectrum of the microbial molecule that you want to deposit, and that should be in the form of a USI. So for those who don't know what a USI means, it's basically a combination of strings that will be linking you to a specific MSMS spectrum. This MSMS spectrum can be, for instance, already in the GNPS libraries, or it can be deposited in a public data set in Massive, for instance, or in other repositories as well. And the way that you can get this USI, it's also shown in the documentation. Oh, sorry, no, CMMC. It's also shown here on the documentation how to get a USI. So there is really step-by-step on how you can get this information. But this is basically what would be a USI, for instance, of a spectrum that is already deposited as part of the GNPS library. So this is basically what we need as a mandatory part in the deposition workflow. So this is the very, very first thing. This MSMS identifier would be pasting here the USI. And then we also have another field here in which we ask the question, has this metabolite been confirmed or predicted to be microbial? Well, here you have the option of putting both. In many cases, for instance, if you have, let's say if you have, of course, isolated the compound from a microbial strain, or if you have, for instance, a monoculture and you have a class of molecules that you find on the culture but not on the media, of course, this has been confirmed to be microbial, so you can put it that. In the sense of predicted to be microbial would be, for instance, let's say that you synthesized a compound and then you did a search in microbe mass, and then you did a search of the MSMS spectrum of the synthetic compound in microbe mass, and then you get the the potential microbes that could be producing this compound. So that would fit in predicted to be microbial. And well, yeah, this, if predicted, why do you think it's a microbial metabolite? You would be able to put this information here. This is just an example. So then we have the second section here, which would be the metabolite identification. Here you can put the molecule name of the microbial metabolite that you want to deposit, the structure in the form of smiles or inchi. Even if you don't know the structure, you can also deposit the spectrum, it's fine. But let's say that you already have information about at least the class that this molecule belongs to. You can also put this information here in the partial structure field. We also have here molecule origin. So basically here is a drop-down menu in which we can say if this molecule is a product of microbial metabolism of drugs, of food molecules, other human-made molecules, which would be, for instance, pesticides and pollutants and such. We also have microbial metabolism of host-derived molecules or of microbial-derived molecules, or host metabolism of microbial metabolites. And also, of course, de novo biosynthesis by microbes, which would be the natural products. And even if you don't know as well, it's not specified, you can also put this information here. And well, if it is, if you know that it's a transformation that is happening, you can also put the name of the original molecule and also the structure of the original molecule in the form of smiles, for instance. And we then have another section called taxonomy/phylogeny section, in which we are going to put the microbial information indeed. Here we can put the NCBI ID for the microbes that are producing the particular metabolite that you are depositing, the microbe name. If you have sequence information, you can also put here in the form of FASTA. And if you are working with a microbial community, you can also put here the link to the raw sequencing data, and you can also put the description. Oh yeah, and also like if you put your cursor in each of these question marks, you'll see a brief explanation of what that field means. And well, also if we have several microbes that could be producing that metabolite, you can put this, you can put all of this information separated by semicolon as well. So the second main the next main section would be the biosynthesis. So here, if it is already deposited in MIBig, you can put the MIBig ID here. If not, you can put the sequence of genes, the gene names and sequence and description. If it has already been published, but it's not in MIBig yet. And also you can go deeper into the description here of biosynthesis if it is already known. And we also have here metabolite source, if it is only microbial, if it is microbial and host, microbial and diet, microbial host and diet. If it's only diet, we are also included that. And if it is also unknown. And for the activity section, this is really an open field. Of course, there are many types of activities that can be reported. So here it's really open so that people can put what they want. But in the future, we expect that we can perhaps restrict a little bit into main sections, at least to be easier to reuse this information later on. And here we would have, we can also input the amounts and units of the observed activities. And also we can put the phenotypes that are associated with the molecules, if it is upregulated or downregulated, and also the amounts and units of the observed phenotype. And well, we have the references. Well, I think this is really, really important because sometimes we access some databases that we see a lot of information, but we can't find references. So I think that this is really, really important. So here you can put all the references that support what you are uploading, right? And you can put in the form of a PubMed ID, but if it's not in PubMed, you can also put links to preprints and such. And we also have here the grant information. So you can put the grant agency and grant number. And we really encourage you to put this information so that we can so that the NIH can also keep track on what are the grants that are contributing to this initiative and so on. And here as the very last field, we have the comments section in which you can put any other additional information that you think it's relevant for this microbial metabolite. And I would like to emphasize here that this workflow, all these fields that I have briefly showed you, they were actually built with the community and with mainly the grants that are associated with the CMMC. Because the very first version of it, we had much less options for each of these fields. And after we had several meetings with them, they said, oh, we are actually working with microbial communities. So then we added the field of microbial communities. So I would like to also let you know that if you feel that there is a specific field that would be needed to be added or a specific option in the drop-down menus, please feel free to contact us and we will do our best to also expand what could be added, what type of information we can add into this knowledge base. So does anyone have any questions for now? Feel free to stop me at any time. Yeah, so here it's so then after you put all this information, you can just submit the workflow. And well, I also want to point out to the documentation that there is very, very detailed explanation on the deposition workflow. And on each of the fields, you will see that more on the bottom. There will be detailed documentation and also examples for each of the fields. So I highly encourage you to take a look into that if you are interested in contributing to this knowledge base. So, well, the question is, okay, where does this information go then? How can we access everything that was deposited? So we can access that. We can access the CMMC knowledge base by CMMC KB GNPS2.org. I'm also going to paste here on the chat. And it's also on my Google slide, on my slides as well that are on the shared folder. So here is how the knowledge base look like. Of course, that here we are just showing some of the fields that can be deposited just for the sake of space. But if you want to have the information of all the fields that were populated, you can just click here on download. We separated this knowledge base into USI database, which is the spectrum database, and also the structure database. So here on the top, you're going to see all the USIs. And on the bottom, you're going to see it separated by structure. This is because, well, we also encourage people to deposit the different spectra that a particular microbial metabolite can be generating. So for a molecule, we can have much more USIs as we are seeing here. So these are probably different ion forms, different spectra obtained from different instruments, different analyzers and such. But yeah, here it's separated by spectra and here it's separated by molecules. And what we also did more recently is to link as well if this compound is also deposited in NP Atlas, for instance. Many of them are. So you already have a direct link into this other database. So right now, I'm going to go back here quickly to my presentation just to show you some numbers here. Right now we have 4,439 unique MSMS spectra that are deposited and 721 unique structures. So this has been the result of a large effort within our lab and within the grantees that are associated with the CMMC and also with the community. Well, a lot of people, I can see some here that actually responded to our call on Twitter. So we also made a call on Twitter in which we're saying, okay, do you work with microbial metabolites? If yes, please contact us. We would love if you could also deposit your information into the knowledge base. And well, this has been very well received. Well, so right now I went briefly into the CMMC knowledge base, but now I would like to go into a little bit of more details on the network enrichment analysis. And well, for that, our test data set will be 2 fecal samples of the American Gut Project that I briefly mentioned before, and also 3 different microbial monocultures in which bile acids were added into the media, and we wanted to see if there would be any transformations happening. So let me go back here. Yeah, so if you wanna if you want to take a closer look into the data, it's deposited here in Massive. It's publicly available, and well. Okay, so this is the original molecular networking job. So basically what we do with the network enrichment analysis, we basically want to enrich the information that we already have from a previous molecular networking job. So this is an example of a job that was running in GNPS1, it's fine, and that we want to enrich. We want to run within this we want to compare the spectra that we have here with the spectra that are deposited in the knowledge base. So this is, as I mentioned, the original data set. Let me click here. If you click in this link, you're just going to, oh, I need to be logged in. You're just going to see that, yes, that there were 2 samples from the American Gut Project here and 3 different bacteria monocultures as well. And also we uploaded the media blanks as well. And well, we want to then enrich this information. Of course, that we are going to, we have the spectral matches here with the GNPS, but we don't have the microbial information that we would like to have. So we can go back to GNPS2 and to the workflows and type again CMMC. I went with you through the deposition workflow, but now we are going to check the network enrichment workflow. And it's a very simple, very simple workflow. So here you just need to input if the network that you want to enrich, it comes from GNPS1 or from GNPS2. That's it. In that case, I'm going to select that it's coming from GNPS1. And then we need to put the task ID. So the task ID is here on this is the original molecular networking job. It's on the Google folder that I shared with you. This is the task ID, this part here of the original molecular networking job. And we can put it here, just paste here. And then we specify if it's a feature-based molecular networking or classical molecular networking. In this case, it was a classical molecular networking job. And well, it's of course that if you have a molecular networking job that was run through GNPS2, you can of course also use that. I'm not gonna show you here right now, but it's also present on the step-by-step on all the screenshots that I have on my presentation. But well, basically what we want to do is always import the information that is already available, like the molecular networks that are already available and the spectra that are available as part of another task. And here we are going to be selecting all the parameters for the library searches. So it's another library, it's a second generation of library search that we are having. We already have the library search that is done naturally with the molecular networking job, but here we are running these library searches within the spectra that are deposited in CMMC. So here we can specify the precursor ion tolerance. I'm going to leave here to 0.2 because it's high resolution data. And the fragment ion tolerance as well, I usually put it as well to 0.02, sorry. And then we can also specify the minimum cosine. I always leave that into the default settings, which is 0.7. And we also have here the setting for library minimum match peak. So how many fragments should be matching between the spectrum that you were from your data compared to the spectrum in the library. And here top results to report per query. So here the default is 1 and I'm going to leave it as 1 as well. But let's say that you want to see if you select here 3, you're going to see the top 3 results for the library search for each node. So you're going to have like, you can also select that you want to have more matches than only 1 for each of the spectrum that you are searching. I'm going to leave it here to 1. And here we have the possibility of doing analog search. Sorry. Here, I'm going to leave it false, but you can also select it as true. And if you want to use the analog search, you can also specify which would be the maximum mass shift as well. But I'm going to leave here to false for now. And then it's just really submit workflow. And then it's going to take about 20 minutes. This is because it has to download the data, the knowledge base, and then do the network enrichment. So it takes about 20 minutes from my experience. But of course that I already have a job that is ready if you want to follow the next step-by-steps and explore the results together with me. It's here on the Google Docs. So this is the job that we are going to be exploring. I'm also going to paste here on the chat. And I'm going to open this as well. This is basically how it should look like after it's done. You see, it took about like 18 minutes to run everything. But before I proceed, does anyone have any questions at this point? Okay, so again, feel free to stop me at any time. Oops, sorry. Okay. So well, the very first thing that we can do is check the CMMC search results. So here we are going to see all the compounds that are part of the CMMC knowledge base that matched with this particular data set. And what is very cool is that we already have all the microbes named that have been deposited as well to be producing this particular compound. So here you're having the compound name, the cosine similarity, all the microbe names, the microbe NCBI, and the spectrum, what is the source, and the structure here at the very right. And well, we can see here that there are some interesting molecules. We can see here some bile acids, which is what we expected, because as I mentioned, we put some unconjugated bile acids on the media. There were some unconjugated bile acids on the media, so we can see here some conjugated bile acids. So this is how you can very briefly explore what would be the compounds that matched with the knowledge base, and then you already have right away which are the microbes that have been also deposited to be producing these compounds. And well, if I click back here to go back to the status page, what we can do then is also explore the networks. So right now, it's not really, you can explore the networks here directly on GNPS2. I know that it has already been described before in the VMOL series, but for what I want to show you right now, I'm going to be using Cytoscape, which is open source software that enables us to explore networks overall. So I'm going to click here on Browse All Results, and there are some specific files that we will need to visualize our network and see like how it was enriched after our analysis. So we can, so by the way, let me go back here. I downloaded everything. It's a bit heavy, it takes a little bit of time, but I downloaded everything. All the results are also here at the Google, the Google folder. So everything is also here. I'm actually going to, yeah. So everything is here. But if you want to download directly here from GNPS2, you can come here to GNPS network, this folder. And then what we want to download is the network GraphML file. So I'm downloading this. And we also need another important file, which will be here on CMMC results. And what we want to see, what we want to download is the CMMC enriched results. So I'm also downloading this. So these are the two main files that are like the most important ones for you to visualize your data. So I'm going to open here Cytoscape. Okay. Yes, done. Let me actually do that. And what we want to do is open the GraphML file that we just downloaded. It's right here. And we can just drag and drop the GraphML file, just download it here to Cytoscape. And this is our molecular network. This is exactly the same molecular network that we have from the previous, from the original molecular networking job. So, but what we want to do as well is to then import the TSV file that we also downloaded right now with the enriched results. So with that, we can come here into import table from file. And then we need to then select the CMMC enriched results.tsv. So right now, what we need to do is click here on query scan, and we select that as the key column. So basically, we are going to be comparing this column here, which has the cluster index information from the enriched results with the cluster index information from the molecular networking job. So it's kind of merging all the two different tables that we have, the table from the molecular networking and the table from the enriched results. So it's basically select query scan and then key and then okay. It's really quick. So now we are going to have all the information that were enriched as well, all here in Cytoscape. So what we can do, let me go here to style, is I'm going to just change some aesthetics a little bit. I'm going to put the nodes as white, just for better visualization. And the border as black and a little bit thicker. And I'm selecting here the view, always show graphic details. So this is how our networks look like. And then we'd like to see, okay, which of these have matched with the spectra from the knowledge base, and what are the microbes then that produce these compounds? We want to have it all, right? So what we can do is come here and fill color, and we click here on this arrow. And here on column, you will see that we have all those fields that were populated that can be populated in the deposition workflow. It's many of them, but what we would like to do is, for instance, select here input name and then discrete mapping. So here are all the nodes that had a match with the libraries, with the CMMC knowledge base, and these are their names. So we can, for instance, select everything here, selecting shift. And let's say that we can right click here, go to edit and edit selected discrete mapping values. And let's say that I want to put all the ones that are microbial as green, for instance. So this is how we can enrich our networks. So all the ones that are colored here as green are the ones that match CMMC libraries. And what is very, very cool is that we can see a very important network here in which many of the matches in which many of the nodes had a match with microbial molecules that are deposited in the knowledge base. So of course that we want to know what are these compounds and what are the microbes producing that. And what is very cool is that we can do that directly all here in Cytoscape very, very quickly. So we have a lot of columns here. I'm going to select here show columns to clear that up a little bit. I'm going to, for now, select none because I just want to show some specific columns to be easier to follow. So we can select here input microbe name, the input name, and my input molecule origin, for instance. So let's say that we want to see what are these compounds. We already have all this information here It's the oxycholic acid. It was classified as microbial metabolism of host-derived molecules. And this is the list of the microbes that have been reported to be producing this molecule. So it's a very, very big list. So you already have all of this right away in the Cytoscape when you are inspecting the results. For it to be even clearer, I'm also going to change here the label. I'm going to select here that we would like to see the input name. So we already have right away here all the names of the molecules that matched already mapped here into each of the nodes. So we have cholic acid derivatives here on the right, on the left, and more here on the bottom. We have the oxycholic acid derivatives. So this is really, really powerful and really, really cool. So if you have any questions, please let me know. You can, as I mentioned, all the information that was deposited in CMMC is imported here when we import the table. So let's say that you can also use those categorical variables, for instance, the ones that we had the drop-down menus that they are restricted, right? So you can also map these, for instance, here. Let's say that I want to see here on the field color, the molecule origin, input molecule origin. I can, for instance, select all of this here and say that I want to color them based like on right click and then mapping value generators. And then we can use here this dark colors map, for instance. So we see, for instance, that all of these here that are pink, they are microbial metabolism of host-derived molecules. But, well, if we go to the other parts of the network, we will see here that the indole-3 lactate was classified as a microbial metabolism of food-derived molecules. And if we go, for instance, to this other one, citrioviridine, it's a compound that it's a natural product, de novo biosynthesis by microbes. So you can also map this more categorical variables as well to be like easier to follow. So here is a dipeptide. It was deposited and classified as a microbial metabolism of food molecules. So, well, as I believe that as you can see, considering the type of this data set, we would be, of course, expecting that there would be more here populated, right? Because it's a microbial monoculture. We also have fecal samples. But, well, we are continuously working on expanding the knowledge base. So this type of analysis, this type of conclusions that we can get can only get better and better as the community and people continue to deposit their the information in the knowledge base, and it will only improve on the future. So this is what I wanted to show. I know this is it's a short workshop, but before finishing, Daniel, I would like to also mention that if you have any questions, if you are interested in contributing to the knowledge base, please contact us. We have also office hours as well. It's also here in our website. We have this tag CMMC workshops and training. This is the previous workshops that we gave as part of CMMC. Feel free to watch them. And we also have here the contact and office hours. Every Wednesday at 9 a.m. at San Diego time. We have office hours in this Zoom link, so feel free to join. It's usually me and Shipei who are at these office hours, and we are really happy to take any questions. And yeah, and I think this is it. Please let me know if you have any questions. Feel free to join our office hours. And yes, I'm going
Awesome.
to pass to Daniel.
Awesome. Thank you so much. This was great. Thanks so much for the nice overview and walkthrough of like the CMMC and how to like actually leverage the enrichment analysis. We will certainly make use of that. I'm sure there's many questions, but before we go to the question and answer session, I just want to say goodbye to everybody who's taking off or who's watching this online later. In 2 weeks, we won't have the VMOL seminar because there is the ASMS conference in Anaheim. And instead, during that week, we will have a in-person symposium the Friday, which we will also stream online. So in case you're not in California, during that time, you can Zoom in. I will post a registration link for the Zoom sign up in a minute. And then, yeah, I hope to see you all the latest in 4 weeks again. Bye-bye.